his
afternoon.
lecturer was like, I don't want to teach you taxonomy. Go learn it from those guys. And so she sent Carlin and I here in the June or July or whatever before the first semester
you know.
for me. And so I got my helper. So I don't know who this skinny guy is who's hugging my wife, but we arrived here in that July of 2000, and this much of the same things looked similar. Like I think they knocked down this building 107 from when we were here, and one of the things when I started asking me if I could do this, and I said, oh sure, I can do that, and I thought I had like some sort of like a digital remembrance of my time here because I sent it to Sherry, but I went looking through all the stuff that I have on Facebook, and I didn't start Facebook until like 2008 or something, so there's nothing there. And then I went looking through all of my like old digital photographs, and I didn't even have like a cell phone until 2010. So I was like, do I have any photographs at all from this time period? And it turns out that Carlin's mom came to visit while we were here and took pictures of us and took pictures of the lab and like whatever else was going on. And so we have those, and we have them, printed out in a box somewhere because I was digging through all old photos and couldn't find anything. And then Carlin's like, I have this. And she had some sort of box full of old photos. And we also have some old photos from the Ely Diatom meeting as well that probably nobody's ever seen because they were never digitized. So they just basically were, old photos in a box that Carlin and I have only seen. This is obviously McBride Hall up here in the corner, and then that's Carlin and I studiously working in front of what was the McBride hood, which is a fan in front of a window. We're definitely not using NAFRAC inside in the state of California. Okay, so this is obviously Carlin doing some work on our time here as alumni, and we would go out every day in the field. We would collect samples and we'd come back and process the samples, and then we'd make slides. So it's somebody's turn to make the slides every week or every day or whatever. And we'd start off each of the mornings with a lecture. So we'd go into the old library building where the projector was, and we would watch a lecture for a group of genera by Gene Stoermer. And then at the end of the lecture, he would say, now go forth and do great things. And then he would sort of like lead us out into the field, and we'd collect samples. And we were supposed to keep track of all of our stuff in a notebook, which I did. Again, I didn't have a camera at the time, which is, if anybody knows me, that's kind of a weird thing. But I just drew everything by hand. And so among the things in my talk, you will see a bunch of my hand drawings from when I was here as a student. And I brought my old notebook along with me with all of my old drawings. And a funny thing Mark reminded me of that I completely forgot is that they kept all of the old, like whatever you did was kept on an old computer, and you can see Carlin's entering some of those on this old computer. And Mark said, oh, we have this old database, and nobody has ever looked at as many of these things and identified them as you and Carlin did. And I was like, I didn't even realize it was a contest, but there's not a lot to do in Iowa in the evenings. So the two of us, after everybody went off to play cards and drink or whatever, went back to the lab and went back to look at diatoms because I was just fascinated by them. So go ahead, go forward. Also found this old picture of our TA, who was Sarah Spalding, with Gene Stoermer. Also never been seen by anybody other than me and Carlin and her mom. But you can see Gene there. Okay, so I'm going to talk a little bit about the work that I was doing when I was a PhD student in Nebraska, which is mostly the record of Foy Lake. And that's, the first project that I ever started working on for Diatoms. And I want to apologize for everybody who came after me as a PhD student in Sherry's lab, because I counted 2,000 diatom slides for my PhD from Foy Lake, just from one lake record for a 13,000 year record. So that's like something like 6.5 year resolution between every one of those samples. And some of them are actually even shorter resolution than that, because they're barbed records, and we actually would count the barbs out by fives and take a sample that was an average of those 5 barbs. So there's a really high resolution record that's there. We published a lot of stuff from it. And there's also some stuff I didn't publish, which is actually kind of a lot, as you'll see. All right, go forward. So here's just some sketches. I'm going to start talking about the work that I did in Foy Lake, Montana. You can see I was trying to keep track of things like the strike and on in the groups and whatever else. And I had this really meticulously organized folder. And I was like, who was this guy? Really, honestly, like, I don't know what everything was completely organized. So you could see like even what page I found the thing on as a reference. I was very, very careful. But I'm going to talk first about why we were looking at Foy Lake. So move forward, please. Okay, so this is just a sketch I made of the core. You can see the upper part, which I've blown up here in this about 11 meter deep record in terms of sediment was a little over a meter of barbed sediments and go back about 2,000 years-ish. And this is the part that people mostly associate with the Foy Lake record when they think about it. But this is actually one, if not the most high resolution analysis for a single site in anywhere in the world from a lake. I don't think anybody's ever counted 2,000 samples from a single lake record. And Sherry was like, you don't have to count every one of these. And I was like, do you even know me? I'm not going to just leave stuff uncounted. So you can see here the age models based off of VARs at the top, and then we have some radiocarbon dates and some very prominent ash layers that we use to define the age model for this thing. And we were largely looking at trying to reconstruct things like drought through the Holocene. We wanted to do paleoclimate reconstruction using this sort of very high resolution record. And we thought it was great because there's basically diatoms all the way throughout the whole record, except for a couple of little intervals where they're at the bottom, where there's below the glacier peak, where basically it was, at that time, it was like a frozen over system probably, and there weren't a lot of diatoms. So this is Foy Lake. This is a bathymetric model of it. It's located just up here, basically in Kalispell, near Glacier National Park. One of my favorite places basically to hang out and do research. A lot of my early research is basically in this sort of Rocky Mountain areas. And these are some of the objectives that we were trying to do. We're just trying to figure out like what's the long-term climate variability in the system. We wanted to look for drought, in incidences of severe drought that happened in the 20th century, and how did that look if you go back through this whole record. And we were trying to figure out what are some potential driving mechanisms for drought in the Rocky Mountains, and basically trying to use diatoms to explore these sort of records. So go ahead, Sylvia. And so one of the things that we tied that record to was specific decadal oscillations. This is just basically a long-term teleconnected process. And there's sort of an established, or it was established as I was working on my PhD, that there's a strong influence of the PDO, particularly in the area where we were looking in Montana. And so there's also a pretty strong relationship between that site and El Nino and La Nina and the Southern Oscillation. And so we were trying to figure out like the periodicity of drought and whether it might be affected by these teleconnected processes. So go ahead. Again, this is Climate Division 1 in Montana, and there's where Foy Lake is located. And you can see that there's a very strong relationship between the PDO, the record of PDO that we have from the ocean, and the record that we have for drought in Climate Division 1 in particular. This is the Palmer Drought Hydrologic Index compared to the PDO for that area. And I don't think you need to be prodded too hard to be convinced that those things are probably related to each other, at least in terms of the timing and the direction and everything else. So we were really just going to look at this transaction. Sylvia. Sorry, so I'm going to talk a little bit about the diatoms and the record of Foy Lake and the lake level change record that we created from it. Go ahead, Sylvia. So one of the things that I've specialized relatively early on, we're looking at diatoms as lake level indicators, and the way that we typically did that kind of before I came, and also I apologize to everybody who's done any plankton to benthic ratios since I arrived on the scene, because I also broke that. But the idea is basically you have planktic diatoms, they're more commonly found in deep water systems. You have benthic diatoms more typically found in shallow water systems, and then you can basically just look at the ratio of planktic to benthic diatoms. And if you have a bathtub, which basically as you raise or lower the water level, it produces a nice consistent signal. It should be basically deeper means more planktic and shallower means more benthic. Like Sylvia hit. Go ahead. Okay, so this is just a simple model of that concept. We typically take a core in the deepest part of the lake. And so benthic stuff mostly lives in the shallow part of the lake. The deeper part of the lake is where most of the plankton is. That's where the core is collected. So if we collected a core in a relatively deep lake system, we would get mostly plankton. And most lakes respond to things like drought by basically just responding in terms of the effect of moisture. So if you could just look at that, the precipitation minus the evaporation and come up with some change in the volume of water, and hit that button again, please. And then under low lake conditions, what happens is all those benthic habitats essentially move closer towards the center of the lake. And then when you're collecting a core, you're collecting it from that same spot. But now under shallow lake conditions, you have a bunch of benthic diatoms, right? So it's really a pretty simple concept. We can use this to reconstruct things like lake level change. And then there's also some chemical transitions that happen because when you evaporate water, it becomes more salty, right? Because the salt is conserved. And so you should expect to see it become more alkaline or more salty or something along those lines as well. And so we should see that relationship. Okay, so this is the two examples of slides from the top part of Foy Lake where we had a freeze core where we just looked at the last century of change. And a year after I started my PhD, I ran into Mark and I had my slides with me from Foy Lake. And I handed him this first slide under normal conditions, what the lake looks like. It's almost all one species. And Mark looked at it in the microscope and then he turned and looked at me and he goes, "Is this a joke?" And I said, "What?" And he's like, "It's all the same stuff." And I was like, "It makes it easy to count." So I got the deep lake record is basically just all one species of diatom. It's just the plankton that's in that lake, just one dominant lake type plankton. And then I have this shallow water stuff conveniently occurring in this sort of drought period in that region. And what we got was this massive transition. It's almost all particular halophylla, right? So just a big change towards saltier conditions, but also this massive change. Well, the problem with that is that Foy Lake is like 40 meters deep. And so how do you get like a tiny little drought that creates this massive change in the diatom record where I basically don't have any plankton in it anymore? And so that was a bit perplexing, but I liked this sort of story because I was like, well, there's the drought, right? Like this works. All right, go ahead, Sylvia. So this is for people who don't believe that I'm doing anything that makes any sense. These are aerial photographs showing you what it looked like in the 50s. The lake was relatively deep, and you can see what it looked like in the 30s. The lake was pretty shallow, but it wasn't 40 meters below where it is today, right? So how do we get this huge transition? So that's a great question. Go ahead and hit that button. This is the record of the Palmer Drought Severity Index or Hydrologic Drought Index compared to the Plankton Diatom Index, which is just basically how much plankton is present in my sample, showing you that there's a pretty clear relationship, or at least I thought there was a nice clear relationship between these massive drought periods and then I just lose all the plankton, right? So this was just part of what we were trying to put together from this record, and I think there's a pretty clear relationship there that we established. Sylvia. Okay, so next, one of the things that happened was Sherry was like, well, you have to pass a comprehensive exam and we don't want to have to ask you questions because I'm just exhausting, right? Like if they're gonna ask me the question, I'm just gonna know all the answers and nobody wants to put up with that. So they just gave me like, why don't you just do a model and try to figure out what was going on with the Foy Lake thing? Like, why is it responding this way? So I said, okay, I just made a three-dimensional model, what the bottom of the lake was, and forward. And then you can artificially raise or lower the lake level if you have a three-dimensional model. So once I had a model of the lake system, I could basically calculate what the plankton habitat area is, what the benthic habitat area is, simply by stacking up little intervals. So I just sliced them into tiny pieces, and you could stack them up, and you can make the window as long as you wanted. So if you want a 10-meter lake, like light penetration depth, you can. And basically all the stuff that's up there that's orange would be the benthic habitat. And then if you're wondering, well, what do you do with the plankton record? What I did is just go like anything shallower than 1 meter probably doesn't have a lot of plankton in it. And so we could make a model of the sort of deep area and the shallow areas pretty easily. And it makes some assumptions. One of the assumptions is basically every part of that surface area is covered, which is probably nonsense. But you know, like you could at least get an estimate of what's going on with this sort of a model. And then I published this paper in 2004, where we talked about this process. And you can see, you can just raise and lower the lake level because it's a model. You can put it as low as you want and as high as you want. And under certain conditions, what happens is instead of having one big lake, we end up with two isolated lake basins, right? So when you lower it enough, basically the plankton gets separated in two systems. the benthic habitats all over the place. And so this is what, where I created a crisis, because if I raise or lower the lake level, you can actually produce more benthic or less benthic. And it does not necessarily have to be in a simple bathtub type model that everybody was thinking of before I got here. But what we thought of is that it's probably a good relationship with the actual mean lake depth. And so forward. So here's what it looks like when you just model the photic penetration under different values for every level that the lake has. And so I made these goofy models that, like it makes sense to me, but not everybody else. But if you just plot it against what does the lake's mean depth look like when you raise or lower the lake level, you get a curve that's pretty similar in terms of the shape. And you can see the lake becomes isolated under different conditions when we raise or lower the lake basin. So once we have it in a 3D model, we can play around with all kinds of things and try to figure out how it works. Did I suck the energy out? You hit the button. Okay, so when you have this really long record from the varb interval, what happens is you can then plot up the what we think of as being the lake level for Foy Lake. And here's that. And so you can see this very big transitions and a couple of these very deep estimated drops in lake level where the plankton became very high. And we tied those back to hit the buttons, Sylvia. We tie those to specific intervals and say, well, that's the lake raising and lowering. And one of the things that's kind of confusing for people is you could actually raise the lake level and get a bunch of benthic stuff to come out because you hit that level where basically there's a bunch of plankton at that level or benthic stuff at that level. And suddenly you reverse the trend. You can go deeper and actually produce more benthic diatoms. And if you want some examples of this, you should see Carlin's poster. She's here. She's giving a poster on some stuff that we did where we looked at some lakes in the Midwest using similar model for this. But if you have a relatively simple lake morphology and you're one of those people like, that's not my lake, mine's not complicated like this, take your lake and then flood it and see what happens, because there's almost always a wetland around your lake, and that is a bunch of benthic habitat area when you flood your lake. So you probably have it. Go ahead, Sylvia. So I'm going to talk a little bit about some of the stuff that came out from this. One of this, so I did some time series analysis, try to see if we could find cyclicity in the data, try to connect it to PDO. And then while I was working on my PhD, also Trisha showed up in our lab, and she started doing some really fancy modeling, ecological modeling, looking at diatom body size and how it's related to climate change. And I just handed her a bunch of my Foy Lake stuff, and I was like, just use this. And she put together some really interesting stuff. So go ahead, Sylvia, while you're dancing around there. All right, so this is we broke the lake record into 3 periods, the middle, early, and late Holocene periods, and looked at the spectral analysis. And if you're not used to using or looking at these things, the peaks, anything that's above the red line is basically significant. This is the 99% interval. One of the things that you can, or 99% confidence interval. One of the things that's nice when you have a really high resolution record is you got all kinds of power in the record, right? So you have the ability to look at these things that are basically really short. And if I'm only looking at 5-year intervals, that means I can look at something that has a 15-year cyclicity and nail it. So you can see all kinds of cyclicity that shows up in these records all the way through, and that's cool. They're kind of fit with what our idea of PDO is. Most of them are in the range of 20 to 60 or 70 years. Those are sort of the PDO time intervals. And the strongest peaks are generally in the 50 to 70-year interval, which is PDO. So hit the button. That tells you that it happens in there, but it doesn't tell you if it happens in time with what's going on. It doesn't show you what happens as you're moving through the system. So not satisfied with that. I did this evolutive spectral analysis, which basically breaks the core into little windows and then runs a sliding window through it the same way I was doing with my lake level curve. And it runs spectral analysis on every one of those little windows, and then it stacks them all up into a 3D structure. Super complicated, but this is the entire record from Foy Lake. And this is the spectral analysis. And what you see is that there's time intervals where basically it sort of turns on and then turns back off and then turns back on. So we have this really interesting process where we can link together PDO, and not just like the fact that it's affecting it, but like when does it affect it? So this was a geology paper that we put out in 2006 after I finished with my PhD. Forward. And then Trisha came along and analyzed some of the Foy Lake stuff, looking at changes in body size of the Foy Lake diatom assemblages. And again, because we have this really strong, powerful record with really high resolution, we're able to look at this thing and look at how well diatoms can predict climate changes about to occur or about to affect the lake system and cause a regime shift. And so we see this really interesting shift in the body size, especially in this interval here. that Trisha found. And if you're interested in that concept, there's a paper there that Trisha put together and then generously included me on. So then I want to talk a little bit more about some of the other things that I ran into. One of the things is that there was there's probably still some unresolved taxonomy associated with this. And then there's a large part of the story that actually has never been told. So I'm going to try to tell you some about it. And I've learned a lot from Gene Stoermer with respect to taxonomy. It's extremely important to try to understand what you're looking at for paleo records in particular. If you don't have the right kind of information, you're not going to basically be able to interpret it. So go ahead, Sylvia. This is that weird botanicoid that's basically in the top part of Foy Lake. And when I published my dissertation or finished my dissertation, this was one of my chapters. I was talking about this thing, which we probably today would just say, oh, that's intermediate. Except for one of the things I noticed is that if you look at these things, which we would probably all call intermediate, I looked at some stuff from Mirror Lake and Beaver Lake and Foy Lake and Curtis Lake. They had different numbers of vermiportula at different sizes. And so I was like, that does not seem like something we should expect to see if these are all the same species. So like, you know, this is 4 when that one's mostly a 2 or 3, is probably not a reasonable assumption that these are the same organisms. So and actually talked about this a little bit in the second Diatom, North American Diatom meeting that I was in Key West or wherever that was, Isla Morada. And then when I finished my talk, everybody was like, I have these things, what are they? And I was like, this is year 3 for me. I don't know anything about diatoms. But I still don't know anything about them. Go ahead, Sylvia. So here is some sketches that I did of this thing with some SEM images. And one of the things that's kind of interesting that sort of set this thing off is it just has a single ring of central photoportula, and nothing else really has that except for the one from Foy Lake. So I thought this is probably a new species. I've never published it. It's just been sitting there along with a bunch of other stuff I haven't published. But it's possible that it's a new species, and we still need to kind of dig into that a little bit. It gets confusing when you start looking at those things. So this is what it looks like. Hey, don't go back one. You're ruining it. No, forward one. Just one. There we go. This is what happens when you count 2,000 diatom slides and then try to plot it up on a graph on a single page. I've had this thing printed on a 6-foot-tall poster, and people were like, I can't see the details. And I'm like, sorry, it's just the way it is. So I'm just going to break it into intervals, but then now you can go just one. This is the part we published the record for. This is the part that's unpublished that Sherry bugs me about constantly and says, why didn't you publish that thing? There's a lot of work here that actually never got published, and I've never talked about it, largely because I'm worried Sherry's going to make me try to write another paper about it. It'll happen, hopefully. Go ahead. Okay, so let's talk about this record in the early part of it. What we see is a bunch of colonization, deglaciation sort of stuff. The whole record is dominated by Cimbella falsa diluviana, sort of a cold weather, low nutrient organism that's in the system. It's mostly benthic. There's not a lot of plankton in the system. And then there's a little bit of these sort of tiny tychoplankton guys that are here, the little fragilarias of like moating and cold water and that sort of thing in these systems in alpine settings. And then there's a transition that happens where the lake system sort of shifts to dominance by plankton. I'm assuming this is because it was a very cold water system and it starts to open up. And then we got deep water sort of showing up in the summertime and frozen over lake system the rest of the year. It's dominated by Fragilaria tenera and some tiny little Stephanodiscus. And when I was trying to figure out what this meant during my dissertation, I was like, I don't know what that means. How do we get Stephanodiscus just showing up? It went from low nutrient to what we assume to be high nutrient system. And then it just disappears. And I was working with Jasmine quite a bit, and we came up with this sort of conversation about how deglaciation sort of affects the phosphorus. And one of the things that happens is after a while, soil starts to develop, and then the soil binds up a bunch of the phosphorus. And so we have this story where we have deglaciation, we have these exposed surfaces, and then basically we have soil that developed in the basin. And once that happens, there's no more phosphorus coming into the lake, and we lock it up, right? So that's a cool story I didn't know. Sylvia? And then if we move into the mid-Holocene, it's actually this really sort of simple story where we shifted to, this is Lindavia michiganiana, and then a bunch of benthic diatoms, and it just basically alternates between phases of high and low lake level. back and forth as we work up towards the surface. These little bands that are here at the top and the bottom are where the diatoms were dissolved out. So I think maybe it reached some sort of a low lake stage and became alkaline, and basically there was no diatoms preserved in those intervals, but they make a nice boundary for each one of these diagrams to kind of split them apart. Okay, so this is the whole record from Foy Lake, which I haven't published, as I mentioned, for most of it. You can see this is just sort of a summary of what we talked about, the lower part of the lake shifting into sort of this stepanidiscus dominated interval. There's a little blip at 8.2, which is important for people who care about connections to teleclimate processes. And then it just goes basically deep, shallow, bounces around until we get to the top part of the lake, and then suddenly it dissolves. And then when we come out of the dissolution zone, it's dominated by just that one plankton. Okay, so I'm going to talk just a little bit about like what we're going to do going forward, hopefully with this, and what I've been thinking about doing with the Foy Lake record, besides just publishing that part. Sylvia, hit it. As a side note, Mark asked me, and we're going to talk a little bit about sexual reproduction in diatoms, but during COVID, I was trapped at home, and I had my microscope with me, and I did a Diatom Web Academy talk about this guy. At the time, it didn't have a name. It was Stepanidiscus sp. And we've since described it and called it Stepanidiscus cruscus. And what I did was, while I was trapped there, I started to notice that the initial valves were visible because there was so many of this one thing in the sediment record. I started measuring the initial valve sizes, and I realized that at the top of the core and at the bottom of the core, we got totally different initial valve sizes. And I was like, that's weird. Like, what's causing that? So did it again, actually. So I plotted them up. And what you see is that there's a really, really strong transition that happens in the size of the initial valve as you go from the bottom of the record about 3,000 years ago in June Lake. And as we move through it, basically, what you see is the initial valves start getting bigger and bigger and bigger. And at the top, they're actually almost double the size of their original one. So I see this as being the evolution of that diatom right before our eyes, as the initial valve is something that is usually fixed. So I want to try to do something like this, probably with the Foy Lake record eventually. Sylvia, hit that. We do find a ton of the initial valves in the Foy Lake record, and so I never thought of doing this when I was doing that, but I want to give a shout out to Virginia Card, who at some point gave a whole bunch of the Foy Lake slides to, because she was looking at these sort of long-term dynamics and evolution and the organismal sort of ecology of these things. And so I think this is sort of an interesting story that we could kind of dig into, and maybe Mark's talk that follows mine will have some more about that. Okay, so that's it. This is the logo that I drew for the Diatom class, for Diatom class of 2000. And on the front, it says the Diatom capital of the world, because I don't know if you've driven around to any of the cities around here, but every one of them is the capital of something. And I feel like Okoboji should be the capital of Diatoms. So, thank you. Great job, Sylvia. Thank you.
Maybe I missed this. What do we call that one diatom that dominates the plankton, the uppermost layer?
This message was redacted. Yet.
We took one more. Hi. Have you considered humans in that landscape? Do you know if there's any history of humans using the landscape?
Yeah, there's people that have houses around it.
No, in the past.
Oh, very ancient people?
Yeah.
There's no evidence that we know of for that, for this particular site that we're aware of. So, okay. Is Sarah allowed to ask a question, or you need to do something?
No, I need to ask a question from the Zoom. So it needs to be on here. Why do you think the diatoms weren't preserved in those two intervals?
Yeah, it's probably just the ionic concentration. So as the lake level lowers, basically it increases the alkalinity, and then diatoms usually don't preserve in conditions higher than pH of around 9 or 10, depending on burial rates. Yeah.
Could that have when the lake level lowered, instead of a calcium base, could it have been sodium base? So it's not alkalinity, but higher. Yeah, higher.
I'm not sure.
Yeah.
Yeah, it could be, but I'm not
Okay, thanks.
All right, thank you.